Chapter 20, Virology flashcard questions. Number 1. Use of bacteriophages to cure bacterial infections in humans. It has seen renewed interest in treating infections caused by multi-drug resistant bacteria called superbugs. Bacteriophage therapy. 2. A complex community of microorganisms that forms a protective and adhesive matrix that attaches to a surface, such as river rocks, artisan well pipes, industrial pipelines, dental plaque, catheters, pacemakers, and other medical devices. Biofilm. 3. Identical subunits composed of structural proteins called proteimmers that form the capsid of a virus particle. Capsomers. 4. A porcelain filter that retained and concentrated non-filterable bacteria but allowed viruses in a liquid such as tap water to flow through. The use of these became a standard practice to remove bacteria from drinking water after 1884. Chamberlain porcelain ultrafilter. 5. An enzyme that hydrolyzes specific bonds in the muron or peptoglycan layer of the cell wall of a bacterium that allows bacteriophages to escape or be released from its bacterial host. Endolysin. 6. An enzyme used by bacteriophages to create pores in bacterial cell walls to facilitate host lysis and bacteriophage release. Holin, spelled Holin. Number 7. An infection caused by the foodborne bacterium Listeria monocytogenes. This infection is a primary threat for pregnant women because it may cause life-threatening complications for the mother and her baby. Listeriosis. 8. When a prophage carries genes that alter the phenotype of a lysogenic bacterium. Lysogenic conversion. 9. An enzyme found in tears and saliva that digests the peptoglycan of gram-positive bacterial cell walls. Lysozyme. 10. An enzyme that hydrolyzes specific bonds in the muron or peptoglycan layer of a bacterial cell wall that allows bacteriophages to escape or be released from the bacterial host produced by some bacteriophages. Muramidase. 11. Research studies used to examine a single replication cycle of a particular virus by infecting host cells with viruses at a high multiplicity of infection. One-step growth experiments. 12. The first European clinical study underway evaluating phage therapy to treat septic bacterial infections caused by Escherichia coli, E. coli, or Pseudomonas aeruginosa. Burn wound infections. Phagoburn. 14. The number of virions it takes to form a plaque on a monolayer of cells infected with a known quantity of viruses. Plaque-forming units. 15. The viral DNA of a bacteriophage that is inserted into the bacterial genome and replicates without producing bacteriophages within the bacterial genome from one generation to the next during binary fission. Prophase. 16. Refers to bacteriophages that undergo a lysogenic replication cycle in which the viral DNA integrates into the bacterial cell's chromosome to form a prophage. Temperate. 17. A toxin whose harmful properties have been destroyed, but that still retains the capacity to produce an immune response. These are used as vaccines. Toxoid, spelled Toxoid. Number 18. An enzyme that binds to the end of a transposon and randomly catalyzes its movement to another part of the host genome using a transposition mechanism. These genes are carried by retroviruses known as integrases and bacteriophages. Transposase.